just let me know.
Let's just recap what we covered last time. In sequence, these are 3 different metabolic pathways that resynthesize ATP. Really, you don't store ATP. There's no storage container, vesicle stuffed full of ATP. It's just in the cytosol. And at any given moment, there's enough already made sitting there to maybe power muscular contraction for a few seconds. One of the graphics in the book showed 6 seconds. That might be pushing it. It's probably more like 2 or 3. But either way, there's no significant storage for it. So right away, you have to start resynthesizing. As quick as you break it down, you have to reform it. And the equation that you want to have in your head to make sense of this, hydrolysis of ATP, and then resynthesis. Now you can't use the energy from hydrolysis to resynthesize it, because once this energy is liberated, it's used to do work. The myosin head is using it. So this energy is gone. So you've got to get energy elsewhere to reconnect inorganic phosphate with ADP and reform that high energy phosphate bond to resynthesize ATP. So this is what we mean when we talk about ATP turnover. As quickly as it's used, it has to be resynthesized, or else you run out. You run out, cells die. So where is this energy coming from?
Creatine phosphate. Creatine phosphate.
Okay, you want 2? What else? Glucose? We should be fair. And? That's coming from glucose. Fatty acids. FFAs, free fatty acids. And we just doubled everything up for good measure. So you store creatine phosphate, you store glucose, you store free fatty acids in the muscle cell. When the muscle cell runs out of creatine phosphate, that's it. There's nowhere else for it to get it. Can't pull it out of the bloodstream. But when the cell runs out of glucose, because it broke down all of its stored glycogen, when it runs out of free fatty acids, because it broke down all of its stored triglycerides, then you can pull those things from the blood. Nutrient levels is a homeostatically regulated variable. Blood glucose levels have to be maintained relatively constant, about 100 mg per deciliter. As muscle cells take glucose out of the blood, who puts it back into the blood to keep the levels stable? It's a digestive organ. Liver. So you have a huge storage of liver glycogen. Now, of these different fuel sources, which one is easiest to break down, meaning requires the fewest steps, reactions, enzymes? Creatine phosphate. So one step, one enzyme pathway. Creatine kinase takes the high energy phosphate that's currently attached to creatine and puts it on ADP. So when creatine phosphate loses the high energy phosphate, it becomes creatine. And when ADP gains it, it becomes ATP. So you just take that high energy phosphate off of one molecule, slap it on another. And that is phosphorylation. You could say creatine phosphate is phosphorylating ADP. It's giving it a high energy phosphate. Next in line, glucose. Why not free fatty acids? Well, two reasons. Glucose you can break down in the absence of oxygen. And when you begin exercise, ATP turnover in the muscle happens faster than oxygen delivery to the mitochondria. In other words, metabolic rate in the muscle goes up in an instant, but the cardiovascular system, heart's going to take some time to increase blood flow to try to match the increased metabolic rate in the skeletal muscle. And we'll talk more about blood flow in 431. But in general, that's the reason why your heart rate goes up when you exercise, to increase cardiac output, the amount of blood getting pumped through the blood vessels per minute, so that you can deliver more oxygen to tissues that now have a higher metabolic rate. The other reason, there's more steps involved in breaking down triglycerides compared to breaking down glucose. Once you get the free fatty acids off of the triglyceride, then you have to cut it up into 2 carbon pieces. And then each 2 carbon piece goes into the Krebs cycle, which is the enzymatic pathway in the mitochondria. Pyruvic acid can enter Krebs cycle. Certain amino acids can enter Krebs cycle. Fatty acids can enter Krebs cycle. So if oxygen is present, you can start the breakdown of glucose in the cytosol, get your 2 ATP, your 2 molecules of pyruvic acid, and then you can continue to further break down what was glucose initially until there's nothing left but CO2, water, ATP, and heat. But if pyruvic acid can't go into the mitochondria, then you don't make the extra 34 ATP. Instead, pyruvic acid is converted to lactic acid, most of which gets picked up by other tissues from the bloodstream, like the heart. So anaerobic glycolysis means you stop here. Aerobic glycolysis means you start here and then you finish in the mitochondria, and you get the full 36 or so ATP, as opposed to just 2. So why do this at all? It's so inefficient. You're barely breaking down glucose. You're only getting 2 ATP. Well, because it's fast. It's about a 10-step reaction to get you those 2 ATP. Not as good as the 1-step creatine phosphate reaction to give you the 1. But you've got lots of glucose. And you don't need oxygen. This is happening in the cytosol, not in the mitochondria. So there's really nothing to slow it down. It can keep up with the metabolic demands of the muscle cell. It's not a problem. The problem is the lactic acid. Because as it accumulates, that eventually will fatigue the muscle. But you can get, a good 30, 60 seconds out of it, of maximal activity, like sprinting. Why convert pyruvic acid to lactic acid at all? Why not just convert it to pyruvic acid and leave it at that? Here's my hint. It goes back to chapter 2, equilibrium reactions. In theory, it can go both directions, but we're going to do something to keep it flowing from left to right. So, never mind that for a moment. What do you do to keep this reaction going from left to right? In this beaker of water, we have some A, we have some B. Some of it's already reacted to form AB. We want to keep making more AB. So what do? Keep adding reactants, right? And we've got tons of glycogen stored in the muscle, so we keep breaking it down, keep providing glucose. But you got to do something with the product too. Can't let it accumulate. That'll slow down the reaction. It's got to be removed. And as long as you keep supplying reactant and removing product, you'll keep it moving. So we convert pyruvic acid to something else. Don't let it accumulate either. It then leaves the cell, spills over into the bloodstream. That keeps the reaction moving. That's the only reason why you even convert it to lactic acid. It just doesn't have a shuttle system. I don't know why it evolved it that way, but it's stuck inside. So if you get out of your chair and you start jogging, walking, sprinting, it really doesn't matter what the level of intensity is. The sequence is always the same. For the first couple seconds, okay, stored. ATP, just sitting there in the cytosol. But beyond the first few seconds, which pathway is the first major contributor to ATP resynthesis? Phosphagen system. It's not like a light switch, like the others are off, and this one's turned on, and we're going to wait and turn these other ones on later. They're all running. And they've been supporting the metabolic demands of the muscle while you've been sitting in your chair. But you're suddenly going to increase the metabolic demands of your muscle. You're going to go from a 2 to a 10. Metabolic rate's going to go way up. You can't get oxygen fast enough into the mitochondria to support the aerobic pathways that you've been relying on up until this point. The cardiovascular system will try to catch up, and eventually it can if the intensity level's not too high. But that's going to take about a minute or so. So in the meantime, you've got to rely on phosphagen system. It's the first major contributor because it's the fastest. It's a one-step process. And then you move on to the 10-step process, anaerobic glycolysis. So just about the time you run out of creatine phosphate, anaerobic glycolysis is now the major contributor. And that pathway is revved up, and it's cranking out ATP. And that'll keep you going. And if this is high intensity, you're sprinting, that's probably where you end. You probably won't go more than 30 seconds to a minute, and anaerobic glycolysis will be the major contributor until you fatigue. Lactic acid is going to play a role in that fatigue. But if it's walking or a light jog, then you'll transition into the aerobic pathways. So after about a minute or so, you go from anaerobic glycolysis as your main contributor to aerobic glycolysis. Cardiovascular system has caught up after this minute or so. There's enough oxygen now being delivered to the mitochondria. The pyruvic acid doesn't have to become lactic acid. It can now go into the mitochondria and get you a lot more ATP. And you won't fatigue because you're not producing very much lactic acid. And then, even better, also around a minute or so, you'll start breaking down more triglycerides and start getting some of your ATP from fatty acids. And the longer that exercise continues, the greater the contribution from fatty acid metabolism, the greater it becomes. So if you're graphing contribution of fatty acid metabolism over time, it's going up, and if you track aerobic glycolysis, it's going down. And it takes about 20 minutes or so to reach the point where fatty acid metabolism begins to outweigh glucose metabolism. And the reason is, it is the preferable source of energy. Per gram of substrate, you get more ATP from fatty acids than you do glucose. And it's preferred because you have way more fatty acids in your body than glucose. You have limited stores of muscle, glycogen, and liver. But think about all the adipose in your body. Got enough to keep you alive for weeks. Plenty of ATP to be made from all those fatty acids. So the key to really maximizing fat burning is to do low intensity, long duration.
why they say it's good to do like a low-key 20-minute light exercise in the morning to kickstart your metabolism?
It's better to be, if you're doing 20 minutes,
Yeah.
the highest intensity you can handle for that 20 minutes would be preferable.
Okay.
Because in the end, even though the nutrient blend might favor glucose because the intensity is high, more of your calories are coming from glucose than fat, the total number of calories that you're going to burn in that 20 minutes is going to be 3, 4, 5 times higher than the low intensity. And so in the end, you might actually burn a lot more fat calories doing the 20 minutes of high intensity instead of 20 minutes of low. So the trade-off is more, do I want to do 20 minutes of high or 90 minutes of low to actually burn the same number of fat calories? And then what that reminded me of is the theory that if you're going to exercise 30 minutes a day, better to do 3 chunks of 10 versus 1 solid 30. And I don't know that it's better in general, but there are pros. And the upside is each time you exercise, metabolic rate goes up, and when you stop, it doesn't go down instantly. It stays elevated, and it gradually comes down over several minutes. The longer and the more intense the exercise, the longer it takes for it to come down. So you might actually, overall, have a higher metabolic rate for a longer period of time by cutting it up into 3 versus 1. Because you're coming down 3 times instead of 1 time. I want to make sure that you have answers to the questions in the study guide. Under anaerobic glycolysis, sources of glucose, what are they? Muscle glycogen, liver glycogen. And you could also put blood glucose, but ultimately the glucose that's entering the blood is coming from the liver. So at first, the muscle cell, the one that's active, whose metabolic rate has increased, will use its own stores of glucose. Run out of that, now you bring it in from the bloodstream. But as you bring it in from the bloodstream, somebody else has to put it back and replace it so the blood glucose levels don't fall. That would be the liver. Right, so you could, 2 or 3 sources, depending on how you look at it. And then under aerobic fatty acid metabolism, same question. This is the very, very last line on page 10. Sources of fatty acids for beta oxidation. And what I keep calling fatty acid metabolism, that's beta oxidation. It's a process that takes place in the mitochondria. Beta oxidation is breaking up that fatty acid into 2 carbon chunks, and with them generating ACoA, which goes into Krebs cycle. And if those details that I just said are not on the study guide, then you don't need to know. Sources of fatty acids for beta oxidation. Start with the cell. Stored triglycerides in the muscle cell. And when you run out of that, where do the cells get fatty acids? Pull them in from the bloodstream. But as cells pull fatty acids out of the bloodstream, somebody's got to put them in to keep fatty acid levels stable. Which tissue is going to put fatty acids into the blood? Adipose. So the 3 sources again. Cell, store triglycerides in the cell, blood, but as you pull it out of the blood, who's replacing it? Adipose. All right, now on page 11, About a third of the way down, Roman numeral 7, skeletal muscle fiber types. What you're going to find is many of the differences between slow twitch and fast twitch fibers comes down to their metabolism, the pathways that they rely upon to make ATP. So a lot of what we've just covered is going to come up again. The skeletal muscle fiber types is a way of classifying muscle fibers. When we say slow twitch or fast twitch, we're classifying based on speed of contraction. That can be measured experimentally with a myogram. And if you go back to what I showed you last class, the myogram for the fast twitch fiber occurs over a much shorter period of time and generates more tension than the myogram for a slow twitch fiber. The twitch is slower, meaning it takes more time to reach peak tension, it takes more time to relax, and so it takes more time for the twitch to occur as a whole. It's longer because it's slower. The twitch for gastrocnemius, fast twitch fiber, it's fast because it's shorter period of time. You reach peak tension quicker, you relax quicker. Notice they're not putting tension on the y-axis, because if they did, the green would be much higher than the red. They're putting percentage of max tension. So they're making it a relative comparison. This is 100% of what the gastroc can do. This is 100% of what the soleus can do. But if we looked at absolute changes in tension, this would go up a lot more. Classified by metabolic pathways used to synthesize ATP. So we get the terms oxidative versus glycolytic. And sometimes we combine the two classifications. So you'll see the term fast glycolytic fibers, slow oxidative fibers. So what does that tell you? Fast twitch fibers rely on glycolysis. They are glycolytic fibers. They store a lot of glycogen. They rely mostly on glycolysis. And specifically, anaerobic glycolysis. How do we know that? Because you're going to find a lot less mitochondria in the fast twitch fiber. You're going to find a lot less blood vessels around that fast twitch fiber. You're going to find a lot less myoglobin in that fast twitch fiber. All of those things tell you that fiber uses less oxygen. So if it's using glucose as the main fuel, but it's not relying on much oxygen, then it's got to be anaerobic. It's got to be anaerobic glycolysis. So fast glycolytic versus slow oxidative. If you remember from basic biology class, college, high school, oxidative refers to oxidative phosphorylation. That occurs in the mitochondria in the presence of oxygen. Starts with Krebs cycle, ends with electron transport chain. Oxygen is the final electron acceptor. You don't have to go back and relearn all that stuff. Just know the terminology, what it refers to. The oxidative pathway refers to what's going on in the mitochondria, in the presence of oxygen. So that name, slow oxidative, tells you the slow twitch fiber relies on aerobic pathways, on the oxidative pathways. And since that occurs in the mitochondria, who has more mitochondria? Slow twitch. And since the whole point of the mitochondria is to use oxygen to make ATP, then it's going to have a lot more blood vessels serving that muscle fiber to deliver that oxygen, and a lot more myoglobin to store that oxygen. Slow twitch fibers have a higher capillary density. They store more myoglobin. They have more mitochondria. And all of that says they rely more on aerobic pathways. Everything I'm telling you, I'll repeat many times, and it's in the study guide. It's on the slide. Combining the above classes into a numbering classification. You've got the type 1, parentheses, slow oxidative fibers. But you can just call them type 1 fibers. And then you've got the type 2b, fast glycolytic. So there's our two extremes. The classic slow twitch fiber, the classic fast twitch fiber. And then I've got one in between called fast oxidative. It's a hybrid. The numbering system is type 2A. It's bigger than the slow twitch. Produces more force. But it relies a little bit more on aerobic pathways than the fast twitch. So we call it fast, but oxidative. Fast oxidative. So it's not as strong as a type 2B, fast twitch. It doesn't have quite the endurance of a type 1 slow twitch, but it's somewhere in the middle.
So is the slow twitch aerobic then?
Yes. And then connect this back to motor units. Type 1 fibers are smaller in diameter and associated with the small motor units. They're recruited first. And then skip down to number 3. Type 2B fibers are larger in diameter and associated with larger motor units. The ones that have neurons with really high thresholds. The ones recruited last. And then the in-betweeners, the intermediate fibers, type 2A fibers, are intermediate in their structure and function. So they would be associated with kind of in-between motor units, medium-sized motor units, between small and large. And we have identified other in-betweeners, another one called type 2X. But the priority for you is be able to compare the two extremes. Before you even concern yourself with fast oxidative, the type 2As, just make sure you've got the type 1s down and the type 2Bs down. Your basic slow twitch, your basic fast twitch. And then lastly, you can classify by color. Red versus white. Which ones are red, slow or fast twitch? And the red comes from the increased capillary density, more blood vessels, the increased storage of myoglobin. And with more myoglobin and more blood vessels, you have more oxygen bound to hemoglobin in the blood vessels, and oxygen bound to myoglobin in the cell, and so it gives you a red color. Myoglobin is just like hemoglobin, a red color when bound to oxygen. White fibers. It's not so much what do the fast twitch fibers have that slow twitch don't. It's what don't the fast twitch fibers have that slow twitch do. I just told you what makes the slow twitch red. What is lacking in the fast twitch, causing it to be pale or white? Less capillaries, less myoglobin, less oxygen. And all of that tells you they rely more on anaerobic pathways. Such a fiber isn't going to be able to generate force for a very long period of time, but it is good at generating force. Very powerful fibers, but poor endurance. So then there's two questions there. First, which fibers are better suited for moderate intensity prolonged exercise? Slow twitch Why? To be better at prolonged exercise, you have to avoid fatigue. If you're fatigable, you're no good for prolonged exercise. So why are these guys fatigue resistant? The slow twitch. How do they make most of their ATP? Aerobically, which means they don't have to generate much lactic acid. Second question, which are better suited for high intensity, powerful contractions of short duration? Fast twitch, why? Why are they better for force? Why do you get a stronger contraction? They're bigger. And power is a combination of force and speed. And not only are they bigger and stronger, they contract at a higher velocity. They shorten faster. So that's good for movements like jumping. And we're saying, why are they good for strong but poor endurance? Well, it's okay that lactic acid builds up, because we're only using them for a short period of time. So it's not an issue. So if you're looking for a muscle fiber that gives you lots of force for a very short period of time, fast twitch is perfect. You're going to be done using it before it fatigues. All right, let's take a break. Return at 18 after. Yeah.
I have a question.